Subscribe to our YouTube channel and press the bell icon to get the latest updates.
It is thought that nearly 12% of all human pregnancies start off as multiple pregnancies, where there is more than one embryo in the womb. But only 2% of these pregnancies are carried to term, with more than one baby being born. More than one embryo develops, with more than one amniotic sac visible, but only one ends up surviving. The others, or typically the other embryo, dies in utero and then is either fully or partially reabsorbed in a few weeks inside the womb itself. This happens in 1 in 8 multiple pregnancies, and many times the pregnant woman does not even know that this has happened. This is known as the vanishing twin syndrome. How and why it happens is a mystery. How do multiple embryos form, and why do most of them disappear? This is related also to our understanding of how identical twins develop. In this video, we're going to explore new findings from the Netherlands that talk about epigenetic signatures common across all identical twins, which remain from conception till adulthood. Using these epigenetic markers, even adults can tell if they were an identical twin when they were conceived and if they had a twin in the womb who vanished. I'm Sandhya Ramesh, and this is Pure Science. First, let's look at what a genome is and what the epigenome is. A genome is all of the genetic data that belongs to an organism. It consists of DNA and RNA and all of the genes. When a gene activates, we say that the gene is expressed. This means that the gene, a part of the DNA, is read and transcribed into RNA. Ribosomes then come into the picture and translate this RNA to proteins that are produced at the end of this process. This is the process of gene expression. These proteins then go on to decide what kind of a cell would be produced and what its function and characteristics would be. An epigenome, on the other hand, is basically like the metadata of a genome and also influences how the genome functions. Epi, the prefix, means on or near or with. An epigenome is a log of all chemical changes that have occurred in a DNA because of interactions with other molecules in the cell. The epigenome modifies how the transcription takes place from DNA to RNA. Parts of the DNA or proteins that aid the transcription process are tagged with some chemical tags that direct how the transcription should take place. A set of all of these tags that modify the transcription process are the molecular instructions which make up the epigenome of a cell. We have skin cells, hair cells, eye cells, heart cells, kidney cells, and nearly 200 different types of cells in our bodies. All of these carry the same genome, which is our genome. But each of these kinds of cell have their own specific, unique and different epigenome that is different from the epigenome of the other kinds of cells. The epigenome can alter the DNA or a gene physically and inhibit the expression of it or make it turn off by making it go silent or unreadable or physically inaccessible. Some will unwind the DNA more, making it much easier to read and transcribe and increase the expression of a gene. All of this is completely normal. Epigenetic changes occur throughout our lives from birth till the day we die. Epigenomes also affect how our environment interferes with our genome. When we drink or smoke, for example, we have made changes to our epigenome chemically, which then goes on to also alter our DNA. And this is not just lifestyle, but also general environment, like where we live, the pollution around us, the climate around us, and so on. Even our social experiences and interactions and our feelings can also help induce epigenetic changes because the feelings we feel are all chemicals. There was a famous experiment on mice where researchers studied the epigenome of mice babies that were nurtured well by their mother and mice babies that were not given any attention by the mother. The latter group, who were more stressed, and the genes that manage stress were modified or methylated. GNA methylation is an epigenetic mechanism that controls gene expression, and it is done by the addition of a methyl or a methyl group to the DNA strand. It is an epigenetic tool that can even fix genes that are turned off. Studying DNA methylation helps us understand how some cellular processes work. Unique epigenetic changes in the form of DNA methylation in response to the world around us is why even identical twins can actually go on to have very different health conditions as they become adults. As they're exposed to the world around them, their epigenetic changes differ from one another. So one of a pair of identical twins could actually go on to become a heart patient at 45, while the other could live a completely healthy, disease-free life until the age of 90. And these epigenetic changes can be passed down just like the genome. So essentially, the childhood experiences of our parents and our grandparents can in fact become encoded in our own epigenome. But these changes are not permanent, they are reversible. As the environment can affect the genome, it can happen both positively and negatively. And we understand very clearly that a very healthy lifestyle with healthy food and exercise can alter the epigenome to our benefit. Studying the epigenome also helps us understand a lot of health conditions like mental illnesses, heart disease, cancer, and so on. Identical twins are monozygotic twins, which means that it was a single zygote or a fertilized cell that then split into two embryos during the early stages of development. How this split happens is a mystery. We've actually never understood it. We know that twins run in families, but that's not for identical twins. It's for fraternal or non-identical twins who are dizygotic. So, in a woman, two different eggs are fertilized in the same pregnancy, resulting in two embryos that grow together. But identical twins are monozygotic and they don't run in families. Identical twins occur with the same rate of incidence across the world, across different cultures. To understand the epigenome of identical twins, these Dutch researchers who came out with this study first analyzed the epigenome of about 6,000 people. The study they performed was an epigenome-wide association study, and in this they compared the epigenome of non-identical twins to identical twins to first identify changes or methylation associated with sharing a womb. Then they noticed that along 834 areas of the genome, identical twins shared epigenetic markers that were also identical. These epigenetic markers were involved in early developmental processes such as cell adhesion, how cells bind to each other, regulation pathways, communication pathways, and so on in the womb. These were focused in certain parts of the genome called the centromere and the telomere regions on the chromosome. These epigenetic signatures were present across all identical twins. The researchers then used this data they had to develop a test that can tell with up to 80% accuracy whether an individual is an identical twin. With this test now, even if a person had a vanishing twin in the womb, now we can determine if they were originally a monozygotic or identical twin. There are some mysteries still surrounding this, particularly in the role of these epigenetic markers. Are these epigenetic changes the result of monozygotic twinning, or is monozygotic twinning the result of these epigenetic changes? Or is it a mixture of both? We just don't know yet. We don't know the details, but we do know that they go hand in hand. Identical twins also have a predisposition for some conditions like certain congenital spinal disorders or also traits like left-handedness. There are also people who are not twins but who are left-handed. So with epigenetic tests like these, we'll be able to tell if such a person was a monozygotic twin at conception and if the trait is unique to such twins irrespective of whether they had a vanishing twin. All of this is conclusive data but is still very early data. It's a big lead in understanding identical twins, how the developmental process affects growth after birth, and how being some kind of a twin can genetically affect us in the world even if we did not know that we were originally a twin.